Welcome back to the Classics of Immunology Journal Club. Today, we are going to review a very important and interesting paper from the 1980s. But before we get started, don't forget to click on the YouTube, like the video, subscribe, scroll down and check out my website where you'll find all the classic papers of immunology on the writing section. So there we go. The authors of this paper are Eckhard Podack and Paula Konigsberg. And it was published in the Journal of Experimental Medicine in September of 1984. And the title is the CYTOLYTIC T cell granules, isolation, structure, biochemical, and functional characterization. So this is a total ball of wax of how T cells can kill other cells. I was trying to tell some people about the difference between antibodies. These are non-immunologists. Difference between antibodies and killer T cells. And so the antibodies, basically, that's the Air Force. They're swimming around all the time and looking for targets to attack. Whereas the killer T cells, they're the Marines. They've got fixed bayonets, and they have to have hand-to-hand combat with their target cell. And this paper today is going to show you how they kill. Just in terms of background, it was 5 years previous to this paper in 1979 when we published in January of that year the paper that first described the ability to clone T cells and keep them growing ad infinitum in T cell growth factor, or interleukin 2, which became interleukin 2. And we predicted at that time, if you go back and read those papers and look at the intro and the discussion particularly, we said, this is going to be a revolution for T cells, because we're going to be able to take individual T cells and make unlimited progeny of those individual T cells so that they are genetically identical. They're like identical twins, except there's millions of them, and even billions, as we'll get into in this paper. And that should allow us then to get from the cellular level to the molecular level. And so 5 years later, this is the first paper. We predicted that we were going to be able to find out the molecular nature of the T cell receptor, the molecular nature of T cell killing, and the molecular mechanisms of T cell help for B cells, because they were not smart enough to figure out how to make their antibodies. And the T cells have to help them do that, which I've always felt was amusing. So that came to pass in the 1980s. The 1980s were the golden era for interleukins. More and more interleukin molecules were identified, isolated, purified. The genes were cloned. Unlimited quantities of these new molecules were available for study, and the field essentially blossomed overnight or during the course of the 1980s, as more and more people realized that things were really happening in this particular field, and things started happening really fast. So this paper I'm particularly fond of this paper, as you'll see. I'll tell you why in a little bit, but just a little bit of background on Eckhard Podack. Eckhard was a biochemist from Germany, and he emigrated. He's just 1 year younger than me, actually. He emigrated to Germany, or to the US, to the Scripps Clinic and Research Foundation in La Jolla, California in 1974. And then he stayed in the US for the rest of his career. And while he was there, then this paper came out in 79. So he's been there just 5 years. And he went there to work with a very famous biochemist whose name was Hans Müller-Eberhard, who was also German, who was the person in studying complement. Complement is something we haven't really gone into in detail here, but there's a series of proteins that ultimately can poke holes in membranes. And the last critical component of complement, they're numbered like 1, 2, 3, 4, 5, is C9. Eckhard Podack in Hans Mueller Eberhard's lab, purified C9 and showed exactly how it worked. And it basically is an individual protein that polymerizes and forms pipes that poke holes in the membrane together with antibody. So he was poised. He was really at the right place at the right time when finally you could get your hands on enough cloned cytolytic T cells to begin to find out, okay, so how do these cells, what are these cells doing? How do they kill other cells? Because obviously by this time, by the late 70s, early 90s, early 80s, killer T cells had become a really hot item because they were thought to be so important for transplantation rejection of allografts and cell-mediated immunity in a very general sense in response to viral infections. And also some people hoped that they could be used to kill cancer cells. Eckhard was the first author on this paper. Paula Konigsberg, I think, was a postdoc, and it was just the two of them on this, and it's a huge amount of work, as I'll get into. In the intro of this paper, in the introduction, they basically state early on, or right at the beginning, is that there's morphological evidence through electron microscopy, really, of that there's granules in these killer T cells. Whereas if you look at other T cells, that say non-killer T cells, like helper T cells, some of them are helper, some of them are killer, and some of them are not. But if you look at cells that cannot kill, you don't see these granules. So that was number one. And people sort of surmised or conjectured that there were cytolytic proteins in these granules that were responsible for the seat of. So people thought that these granules were really the seat of where the capacity of killer T cells to kill resided. So in order to get at that question, they thought that they needed to do the critical experiments, which were to isolate these granules from the rest of the cellular components and test these granules themselves for their ability to kill other cells. So that's what they did. In the materials and methods in this paper, in terms of the cells that they're used, there are several different kinds of cells. And this is the part that I like the most, because the cell that they used to do the yeoman's job in this paper to identify and isolate these granules and so forth was the good old CTLL2, which was our first, well, it's really the second T cell line that we generated back in the late 70s, and then cloned it and so forth and so on. And the reason this is important because they could grow unlimited quantities as long as they had a source of conditioned medium to serve as the growth activity, the T cell growth factor activity. And in order to do that, they made their conditioned medium from rat spleen cells that were stimulated with concanavalin A. You know, I've got my cat here. I'm going to have to put him out. It's a her cat. So they used rat spleen cells, and actually this was our recipe that we had generated in order to make T cell growth factor containing conditioned medium. The rat spleen cells were 5 times as big as mouse spleen cells, so that we could get 5 times as many cells from one rat. Therefore we could generate more conditioned medium. And the second cell line that they made was a cytotoxic T cell line, just like CTLL2, that they made after a mixed lymphocyte culture between 2 different allogeneic strains of mice. And at this point in time, the CTLL2 had been in culture for 5 years. And then they made another line they had maintained in culture for 5 months. And then finally they had a bunch of tumor cell lines that did not kill as negative controls in these experiments. So in order to generate granules and to purify the granules from the cells, they grew up around 10 to the 9th cells. So that's 1 billion cells. That's a huge pellet in the bottom of a centrifuge tube. that was our starting material. And that took a lot of liters of conditioned medium and so forth, and meticulous care to grow up these. We never approached 10 to the 9th cells. This was really a tour de force as far as I can see. But that's critical, and it couldn't have done any of these experiments unless they had these cells. So they would take these cells and they wash them a couple of times and so forth, and then they would lyse these cells, disrupt the plasma membranes, so that they would release their granules that then they could purify. So they essentially lysed these cells. They disrupted the plasma membrane with a process called nitrogen cavitation. They couldn't use detergents, which is the sort of the routine way to lyse cells, is to use different kinds of detergents that would disrupt the plasma membrane, because then it would disrupt the granules. So they had to perfect the method of nitrogen cavitation to blow open these cells and allow then all of the cytoplasm would come out of the cells, and then they would spin out just a low speed spin. They'd spin out the nuclei, and they take the post-nuclear supernatant, and they would load the supernatants on Percol gradients, and then spin them pretty fast in a fast centrifuge. And they would get out of these, then they would fractionate the gradient after that. They take 2 ml fractions for about 15 or 20 different fractions, and then test each fraction for 3 things: for cytolytic activity, for protein, and for electron microscopy for morphology, to see whether or not which fractions had the granules and so forth. So then after as you'll hear, they were able to isolate and purify granules. Then they wanted to make an anti-serum to identify these granules that they could then use on immunofluorescence in different cell populations to see whether or not the cells really had granules in them or not. And so they used rabbits that they injected with every other week with 50 to 100 micrograms of purified granule protein. After they generated the hyperimmune sera, they used that to stain granules, as I said, primarily in using immunofluorescence, but also in cytospin preparations. So then they also wanted to test for cytotoxicity for their cytolytic assays, and they used 2 different target cells for this. They used sheep red blood cells, because all you have to do is then, in order to lyse a red blood cell that doesn't have any nucleus, you just have to poke a hole in the membrane and you can lyse the red cells. And then it's an easy assay, because then you just look for released hemoglobin, which you can measure in a spectral photometer at a particular wavelength. So that was easy. And then they also did a chromium 51 release assay using tumor cells, and the same kind of thing, adding purified granule fractions into chromium labeled tumor cells to see if the granules would lyse them as well. And then they also once they figured out that they could purify the granules, they wanted to purify or identify whether or not those granules had any different kinds of proteins in them and so forth. And so again, they made up another billion CTLL2 cells. This time they disrupted they lysed the cells with a detergent, 5% sodium deoxycholate, and then they treated the lysate with DNases to digest the DNA and so forth, and then they loaded the result onto Cephalose molecular sieve columns, took 5 ml fractions from those columns, and then did SDS polyacrylamide gels of each of the fractions to see what size proteins were in those fractions. So in the results, and now I want to do some screen sharing. So this is figure 1. And in the A and B up there, this is CTLL2. Isn't it pretty? This is so a lower power magnification, an electron microscopy. The G0 are Golgi apparatus, and the G's here are the granules. And then this is a higher magnification of the granules, and you can see that they have a dense. This is really electron dense in the center of these, and then here's an even a closer up version of this where you can see small vesicles on the periphery, and then there's a bilayer membrane on these cytotoxic granules. So on figure 2, where they did killer assays on the fractions, and also did morphology on these fractions and protein concentration. And so in the upper panel here, we have results with CTLL2, and in the middle panel is B6, and then, and down here is EL4, which is a thymoma, essentially, that does not kill. So you can see here in the upper panel with CTLL2, the open circles are cytotoxicity, and the solid circles is protein determination. So you can see right here, there's an electron dense, or I mean a dense peak here of cytotoxicity onto sheep red blood cells here. And here are the protein determinations. So there is a peak here with the granules, but it's a smaller peak than here you see on number 2 and number 3. Now these are in the next one of the next figures, we're going to see polyacrylamide gels of the different proteins at 1, 2, and 3. So, and then this is the B6 cell line. And again, and now they tested on 3 different target cells for cytotoxicity, on erythrocytes, on a cell line called S194, and then on another cell line, YAC1, which is a characteristic natural killer cell target that's used. And you can see that there's cytotoxic activity in really the same fractions off of this column or off of the gradient. And here there's a protein peak there, and then another peak that's not as big as this one in CTLL2, and then it goes off and that sort of thing. And now here with EL4, there is no cytotoxic. There are no fractions that are cytotoxic onto the E rosettes, onto the sheep erythrocytes. So in figure 3, as I said, those peaks 1, 2, and 3, and this is non-reducing conditions, and this is reducing conditions. And the only thing I want to point out is peak 1 that had the cytotoxic activity. There are 6 different protein bands that are not present in these other peaks and are not present in non-cytotoxic cells. So this really is what they were looking for here. And they could estimate based on the densitometric scans of these bands here that the granules had to be about 90% pure at this stage of the game. If we go on to figure 5 now, I'm just going to skip, they talk about the purification of these granules and so forth here. So we go on to figure 5, and now we have the CTLL2 target, the B6 cells and the S194 cells. This is in the upper panels, it's phase contrast of the, these are cells now under the phase contrast microscope. And then this is immunofluorescent staining using the rabbit antisera to the granules. And you can see that they are present here in CTLL2 and in the B6, but they're not present in their negative control, which is S194. So then in figure 6 is our depictions of the concentration dependency of the cytotoxicity in panel A. As you increase the granules, you get increasing cytotoxicity. And then this is the time course of cytolysis. And you can see at 37 degrees, it happens really fast. It's basically over within about 2 minutes. Whereas when you lower the temperature, you lose the cytotoxic capacity of the granules. And this is showing the calcium dependency, which was one of the only biochemical mechanisms or assays that you could use to see whether if you could stop it by taking away the calcium from the medium of the cultures, you would stop the cytotoxicity. And so that was all good. So that was figure 6. And then in figure 7 these are isolated membranes from sheep erythrocytes mixed with the granules. And you can see that the granules have basically poked holes in these membranes. There's 2 different size granules or holes. These are 160 ancroms in diameter, and then these are only around 50 to 90 angstroms in diameter. So that's it. There's more data in this paper, and if you're interested, you should read it. But that's all I want to go into today, because it was a groundbreaking paper for this particular field because now they knew where the money was. So let's stop our screen share. And in the discussion of this paper, they lead off by saying this is the first study and the first demonstration of cytolytic activity of isolated granules. Nobody had ever done this before. And as far as they could tell, based on their experiments and so forth, and the fractionation of the cytoplasm, that the granules are the major part of the entire cytolytic mechanism capacity of the killer T cells, killer cells. And they had done different experiments, and they could show that the specificity of the granules were not antigen specific. They were not immunologically specific. They'd kill anything if you give them half a chance, you know? And so the specificity of a killer cell to kill a target cell then had to be, they figured that it was inherent in the T cell itself, the T cell membrane. And of course, the molecular aspect of that is the T cell antigen receptor. That's what you need in order to kill specifically. So it's a combination of the non-specific killing of using the granules, but the target cell specificity is really dictated by the T cell receptor. They go on to show in this paper, and I didn't show you the data on that what happens is that one of the proteins in these granules polymerizes into multi-components and forms a pipe, just like the C9 mechanism of antibody-dependent complement-mediated lysis. That's why Eckhard Podack was right in the right place at the right time, because he was an expert in these kinds of proteins and the assays necessary to really demonstrate how they worked in their activity and so forth. The other thing they say in their discussion is that, you know, this might not be the total story, because it could very well be that there's other things in these cytolytic granules like proteases and DNases and RNases and all kinds of things that could be injected through the pipe once the pipe goes in the membrane that will then digest the cell from the inside out. And that's exactly what happened. There's a whole nother series of papers that deal with the so-called granzymes, which are the enzymes that are in the granules. Because they had never been described before, they gave them a new name, called them granzymes. And there's a whole nother series of papers that if you're interested, you should read those too, because they're all part of the whole mechanism. So what happened subsequently after this paper was published was that Podack, he went right on to the nitty gritty here. He went right on to the money, and he went on to purify what proteins had been named in perforins. He purified and characterized what became known as perforin 1. And that came out in a paper in PNAS in 1985, so just 1 year later. And then he went on to identify and isolate and clone the gene encoding for perforin. But that took him 4 more years and it wasn't until 1989. So from 79, when we first got cloned T cells, to 89, when we really had nailed, or not we, I mean, the field had nailed the mechanism of T cell cytolysis or killing. And then finally, into the 90s, and we'll get there eventually, I think. It's going to take a while. It was shown that perforin, as well as the granzymes, are all interleukin-2 induced genes. And the interesting thing to me is that when we first we made cytospins of our CTLLs when we first grew them. What a cytospin does is that you can take just this very small amount of supernatant and spin it in this little centrifuge, and it will flatten out the cells onto a microscope slide, so that you can then stain it with the right Giemsa or any kinds of stains that you want. So we did that, and we looked down the microscope the first time, and the cytoplasm of these cells was chock full of holes. There were holes in the cytoplasm now, and I've seen a lot of T cells under the microscope, and I never saw a T cell that looked like that. And I was worried at the time. I said, I'll bet you those cells are macrophages because they got all these holes in them. And of course, unbeknownst to me, the fixation process was known to, they use alcohol, methanol, kinds of things to fix the cells down onto the microscope slide. It'll wash out different kinds of things, and it washed out the granules. Whatever was in the granules was gone. There's just holes, but there were a whole bunch of them. They were chock full in these cells, and this turns out was a good thing. And that fits with the idea that these cells are being grown in IL-2, TCGF, for the duration, and they just sort of constantly and consistently made more and more granules. This is a good thing if you want to find out what the granules are and what they do, and so forth. So that's how that story unfolded. And so don't forget to check out the other papers that are on the website that have to do with this. I don't think we're going to go back to the things. This, to me, was the breakthrough paper. If you like this video, don't forget to like it on the YouTube site, and come around to the website and check out the other papers, and then also look at the social media sites. Click on the bell and get notified for more of these videos. I also, in addition to the Classics of Immunology Journal Club, from time to time, I also do things like, for example, on COVID. And last week we made a new video on Omicron, if you're interested in that. Thanks so very much, and come back next week.